Good afternoon. First of all, thank you for the introduction, and I really thank the organization committee for kindly inviting me to this meeting. So let's see. I'm pretty sure that everyone is waiting for lunchtime, so I make sure to finish in about 10 to 15 minutes. Okay. So I have nothing to disclose, and first I would like to acknowledge my colleagues are here at the USC, especially Dr. Jun-Mook Liu. So our laboratory is investigating a molecular mechanism of how do cells become neurons. So this is actually really important because especially in a neuron or any type of cells, fate decision is actually play a very important role to become a proper organs. In our case, it's actually brain. And so for here in neurodevelopment, we have identified a couple of important cell fate determinants. That's including Wnt signaling, Notch signaling, and now epigenetic mechanism. However, the mechanism of cell fate determinants are not really well known as of this point. And it's really important for us to understand those detailed mechanisms in order to facilitate neuron development or neuron differentiation, which can be actually utilizing regenerative medicine. So in our lab, and our colleague and I actually discovered there's one molecule called RIC, which is atypical tyrosine kinase, which is a Wnt co-receptor that plays a very important role in neural outgrowth as well as developing or into cortical neurogenesis. So while we were actually studying for RIC, we actually tried to find what is the RIC's interacting proteins. So on the process, we discover that there's one interesting molecule called Smek, which is suppressor of MEK. So once we found out a couple of good candidates, we pick actually Smek because we tested a couple of times, and every time that we tested, actually Smek is the protein that coming into the higher grade as a strongly bound protein with the RIC. And once we figured out to study this molecule, we found out through Drosophila or Drosophila model that Smek1 is actually a regulatory subunit of phosphatase, which is PP4C. So we actually initially tried to see if in mammalian model, Smek1 is also work as a components of phosphatase. So we did East2 hybridization and immunoprecipitation assays, and we found that indeed this molecule Smek1 is indeed bound to a RIC as a RIC interacting protein, as well as PP4C, which I will going to introduce you later. And then based on bioinformatics on mouse and human, it turns out that the Smek1 mRNA is expressed in the central nervous system, including in the brain. So, but then so far in the existing literature, we haven't found any information on whether this part of phosphatase will actually play important role in neurogenesis. So we start to investigate whether this protein actually play an important role in brain development. So the first step, what we did was to take out the developing mouse embryo, and I tried to take a look at it to see if Smek1 is expressed in the brain or developing embryo. And indeed, it turns out that as soon as the neurogenesis started to happen around the E11.5 in the mouse development, you will start to see the Smek1 expression, especially in the neuroepithelial layers where the new neuronal progenitor cells actually start to born. And around E14.5, where the neural differentiation or neurogenesis peaked, we found out that indeed Smek1 is actually start to express also in a fully differentiated neuron as well. Okay, so so for molecular biologists, what we normally do after we discover that maybe that this protein play an important role in neurogenesis, we try to knock down or eliminate the expression of this protein. So utilizing shRNA or siRNA, we depleted or eliminated the Smek1 expression in neural stem cell and then see what happened. So we found out that if we knock down the Smek1 expression, it turns out that in a cell, in vitro cell culture system, actually the production of a neuron actually declines significantly. This is actually after this 6 days after the differentiation. So we also investigate whether this Smek1 play an important role in early neuron differentiation. So we also investigated to see the number of neurons in 1 day after the inducing differentiation. And indeed, actually even in an early neuronal differentiation step, actually Smek1 play a very important role in production of TUG1 positive neurons. So encouraged by the result, we produced a knockout mouse model to try to validate the phenotype. And indeed, Smek1 knockout mouse resulted in somewhat increase in the proliferating cells, but reduced neurons, as you can see from a picture here. So this is actually the quantification of neuron in an in vivo model. TBR1 is actually the marker for mature neuron. And it turns out that the number of neurons actually significantly decline in Smek1 knockdown mouse. So again, in Drosophila model, we know that Smek1 is a part of phosphatase complex in the PP4C. So we try to see if we can validate that Smek1 really also a part of phosphatase PP4C. And indeed, series of biochemical experiments shows that indeed Smek1 is a part of PP4C, as they bound together tightly. And then also PP4C depletion itself really affected neurogenesis by reducing neuronal marker, which is actually similar to a Smek1 depletion. And then at the same time, we will actually try to see, okay, since this is a phosphatase, that means it should have some sort of a target protein which will dephosphorylate the protein. So we actually utilize mass spectroscopy to identify a couple of candidates, and then it turns out that PAR3 is actually the binding partner, and then we validated the result in using immunoprecipitation that indeed Smek1 and PP4C in our mouse system actually dephosphorylate PAR3. And then, so afterwards, we try to see, so Smek1 together with the PAR3, what is the effect in our mouse system as well as the in vitro culture system. So indeed, PAR3 depletion, PAR3 is actually, we believe that it is actually part of downstream of this whole entire signal system. So we try to see if PAR3 depletion will actually, or deactivation, of PAR3 will enhance the neurogenesis. And indeed, PAR3 actually inactivation will increase, resulted in an increase in TUJ1, which is actually the marker for neuron. And in addition, we also try to determine to see if Smek1 acts downstream or upstream. And then our result actually showed that Smek1 actually acts upstream of PAR3 to negatively regulate neurogenesis. So we actually show, I think I didn't have much time to talk about in detail, but here this picture actually shows that in the presence of PAR3, Smek1 knockdown reduced the number of neurons. So as you can see from this staining, the pinkish actually staining is the marker for neuron. So if you knock down Smek1, then you will see that the reduction of a neuron. However, if you overexpress Smek1 in the presence of normal PAR3, then you can see that the number of neurons actually increase. However, in the absence of PAR3, as you can see, even if you knock down the Smek1 or overexpress Smek1, you do not actually see the increase or decrease in the number of neurons significantly. So we determined that Smek1 really actually act as upstream of PAR3. Okay. And then, actually, this is almost like an end. So while we investigate the expression localization of the Smek1, we also found some interesting fact that during a cell cycle, as we know, the cell cycle consists of a G1, G2, M phase, and S phase. So normally, Smek1 expressed highly in the nucleus. So people think that this is actually a nuclear marker. However, during an M phase, some cell actually turns out that if you can take a look at it in the in vivo study as well as in vitro, that during an M phase or mitosis, somehow Smek1 expression will actually, Smek1 protein will actually flow out from nucleus into cytoplasm. So we are currently investigating, and then also another interesting factor is that during a metaphase and early anaphase, you will start to see that Smek1 actually highly expressed in the mitotic spindle. So we are thinking that maybe Smek1 also may play a role in the controlling of mitotic spindle. As a result, it may probably cause alteration in the symmetrical and asymmetrical division. Okay. So here, this is a summary. So Smek1 promotes a neuron differentiation and as a part of phosphatase. And then also, we found out that the Smek1 really mediates part 3 dephosphorylation. And then also, as a result, Smek1 negatively regulates part 3 in neurogenesis. Okay. So thank you so much. So if you have any questions, please let me know. Okay. So that.